we're going to discuss about the cytokines. So, cytokines are small soluble proteins that regulates the immune system, orchestrating both innate immunity and the adaptive response to infection. These chemical messengers, produced by several different types of cells, have activity modulating effects on the hematopoietic and immune system through activation of cell-bound receptor proteins. Cytokines are induced in response to specific stimuli such as bacterial lipopolysaccharide, flagellin, or other bacterial products through the ligation of cell adhesion molecules, or through the recognition of foreign antigens by host lymphocytes. The effects of cytokines in vivo include regulation of growth, differentiation, and gene expression by many different cell types, including leukocytes. These effects are achieved through both autocrine stimulation, which is affecting the same cell that secreted it, and paracrine, affecting a target cell in close proximity activities. Occasionally, cytokines will also exert systemic or endocrine activities. Individual cytokines do not act alone but in conjunction with many other cytokines that are induced during the process of immune activation. The resulting network of cytokine expression regulates leukocytes' activities and lead to the elimination of the infection. The cytokines cascade produce a spectrum of activities that lead to the rapid generation of innate and adaptive immune responses. In fact, the ability or inability to generate certain cytokines pattern often determines the outcome and the clinical course of infection. In extreme circumstances, massive overproduction and dysregulation produces a cytokine storm that lead to shock, multi-organ failure, or even death, thus contributing to pathogenesis. Initially, cytokines were named based on their activities and the type of cells from which they were first isolated. The major cytokines family include tumor necrosis factor, interferons, chemokines, transforming growth factor, and colony-stimulating hormone, factor. There are also interleukins, which currently number from 1 to 32 interleukins. The interleukins are unrelated cytokines that must be satisfy three criteria in order to be classified as interleukins. They must have their gene clone, they must be inducible in leukocytes, and their biological activities in inflammatory processes must be cataloged. Cytokines were originally thought to act solely on cells of the immune system, but it soon became apparent that many also act on cells outside the immune system. The pleiotropic, having many different effects, nature of cytokines activity relates to the widespread distribution of cytokine receptors on many cell types and the ability of cytokines to alter expression of numerous genes. Many different cytokines may share properties that the activity activates some of the same pathway in genes. This redundancy may be explained by the fact that many cytokines share receptor subunits. For instance, interleukin 6 and 11, leukemia inhibitory factor, oncostatin M, ciliary neutropenic factor, and cardiotropin all utilize the GP130 subunit as part of their receptor. The specific for each cytokine is contained in the other subunit in the respective receptor is generated by the GP130. Thus, some cytokines may have overlapping effects and may alter the activity of many of the same genes. However, redundancy of cytokine activities cannot be explained entirely by shared receptor subunits. Tumor necrosis factor alpha, interleukins 1 and interleukin 6 exert many of the same biological activities, but each utilize a distinct receptor. Here, the cytokines are activating similar immune response pathway through different signaling pathway. Illustrates some of the different actions of cytokines. The increasing clinical usage of cytokines antagonists and cytokine receptor antagonists in conditions such as rheumatoid arthritis, psoriasis, asthma, Crohn's disease, transplantation, and cancer treatment will drive demand for cytokine assay in the clinical laboratory. The pattern of cytokine expression can also determine whether the host will be able to mount an effective defense against and survive certain infections. In addition, numerous crucial part in recovery from infection. The main function of the innate immune response is to recruit effector cells to the area. Cytokines involved in triggering this response are interleukin-1, tumor necrosis factor alpha, and interleukin-6, chemokines, transforming growth factor beta, and interferons alpha and beta. The function of each is discussed here. No. Immunodeficiency syndrome and leukemia are caused by defects in cytokines or their receptors, signal transduction circuits. Genetic and proteomic analysis of this defect will occur within the clinical laboratory in order to assess treatment modalities, effectiveness, and potential gene replacement therapies. We're going to discuss about the cytokines in the innate immune response. So, cytokines involved in the innate immune response are responsible for many of the physical symptoms attributed to inflammation such as fever, swelling, pain, and cellular infiltrates into damaged tissues. The innate immune response is non-specific but occurs within hours of first contact with microorganisms. It may play a crucial part in recovery from infection. The main function of the innate immune response is to recruit effector cells to the area. Cytokines involved in triggering this response are interleukins-1, tumor necrosis factor alpha, interleukin-6, chemokines, transforming growth factor beta, and interferons alpha and beta. The function of each is discussed here. So first, we must discuss the interleukin-1. So the interleukin-1 family consists of interleukin-1 alpha, interleukin-1 beta, and interleukin-1 RA or interleukin-1 receptor antagonist. Interleukin-1 alpha and interleukin-1 beta are pro-inflammatory cytokines produced by monocytes and macrophages. Interleukin-1 production may be induced by the presence of microbial pathogens. Bacterial lipopolysaccharides or other cytokines, interleukin-1 alpha and interleukin-1 beta exhibit the same activities in many test systems and share about 25% sequence homology. However, interleukins-1 alpha remains intracellular within monocytes and macrophages and is rarely found outside this cell. Interleukin-1 alpha can be released after cell death and can help attract inflammatory cells to areas where cells and tissues are being killed or damaged. Interleukin-1 beta is responsible for most of the systemic activity attributed to interleukin-1, including fever, activation of phagocytes, and production of acute phase proteins. It is cleaved intracellular to an active form that is then secreted by monocytes. Interleukin-1 as an endogenous pyrogen and induces fever in the acute phase response through its action on the hypothalamus. The hypothalamus acts as the thermostat for the human body, and interleukin-1 set the thermostat at a higher level. Elevated body temperature may severe to inhibit the growth of pathogenic bacteria and viruses, and also increases lymphocytes activity. Additionally, interleukin-1 induces the production of vascular cell adhesion molecule as well as chemokines and interleukin-6. These chemokines and cell adhesion molecules attract and assist leukocytes to enter the inflamed area by a process known as diapedesis. Interleukins-1 also induce the production of colony-stimulating factor in the bone marrow. thereby increasing the available number of phagocytic cells that can respond to the damaged tissues. Interleukin-1 receptor antagonist is also produced by monocytes and macrophages. It acts as an antagonist to interleukin-1 by blocking the interleukin-1 receptor and limiting the availability of the receptor for interleukin-1. This helps to regulate the physiological response to interleukin-1 and turn off the response when no longer needed. Now let's go to the tumor necrosis factor alpha. The tumor necrosis factor alpha were first isolated from tumor cells and were so named because they induce lysis in the cell. Tumor necrosis factor alpha is the most prominent member of the tumor necrosis factor superfamily, which consists of at least 19 different peptides that have diverse biological functions. Tumor necrosis factor alpha exists in both membrane-bound and soluble form and cause vasodilation and increase vasopermeability. The soluble form is derived from the membrane-bound form by proteolytic cleavage, with tumor necrosis factor alpha converting enzyme. However, the soluble form is unstable and has a short half-life. Membrane-bound tumor necrosis factor alpha can mediate all the cytotoxic and inflammatory effects of tumor necrosis factor through cell-to-cell contact. The main trigger for tumor necrosis factor alpha production is the presence of lipopolysaccharide found in gram-negative bacteria. Tumor necrosis factor alpha secreted by activated monocytes and macrophages can activate T cells through its ability to induce expression of MHC class II molecules, vascular adhesion molecules, and chemokines in a similar manner to interleukin-1. This action enhances antigen presentation and activates T cell to respond to the pathogens that trigger the initial inflammatory response. However, when secreted at higher level, tumor necrosis factor can have deleterious systemic effects, leading to septic shock. This condition results from large amount of tumor necrosis factor secreted in response to gram-negative bacterial infection, causing a decrease in blood pressure, reduced tissue perfusion, and disseminated intravascular coagulation. The latter may lead to uncontrolled bleeding. Tumor necrosis factor alpha and interleukins-1 are both present in rheumatoid synovial fluid and synovial membrane of patients with rheumatoid arthritis. Studies with anti-tumor necrosis factor alpha and anti-interleukin-1 demonstrate that tumor necrosis factor alpha is the central mediator of pathological process in RA or receptor antagonists and other inflammatory illness such as Crohn's disease. Tumor necrosis factor receptor 1 is constitutively expressed on most tissues bind-soluble tumor necrosis factor alpha and is the primary mediator of tumor necrosis factor alpha signal transduction in most cell types. TNFR2 is usually expressed in epithelial cell and cell of the immune system and is activated by the membrane-bound form of TNF-alpha. Overall, TNF-X activity is at least partially regulated by soluble form of both TNF receptor. This act to bind exists tumor necrosis factor alpha and combined with the short half-life of the soluble form severe to limit the cytokine signaling activity. Now let's talk about the interleukin-6. Interleukin-6 is a single protein produced by both lymphoid and non-lymphoid cell types. It is part of the cytokine cascade released in response to lipopolysaccharide and plays an important role in acute phase reaction and the adaptive immune response. Interleukin-6 is expressed by a variety of normal and transformed cells, including T cells, B cells, monocytes, and macrophage, fibroblasts, hepatocytes, and keratinocytes. Astrocytes also, vascular endothelial cells, and various tumor cells. interleukin-1 primarily triggers its secretion. These pleiotropic cytokines